Right, good afternoon. On behalf of the Biochemical Society and Portland Press, I am pleased to welcome you to this webinar, which is part of our Biochemistry Focus webinar series. So topics in this series include different research areas in the molecular biosciences, as well as some practical sessions to support career development. And each webinar will give you the opportunity to ask questions via text, and we welcome any suggestions for future topics and speakers to feature in our webinar series. Please take a look at the website for more details. So I'm Helen Walden, Professor of Structural Biology at the University of Glasgow, and it is my pleasure to chair today's webinar that's titled Structure, Dynamics and Function of the 26S Proteasome. So the 26S proteasome is a very complex ATP-dependent protease, which is required to selectively degrade ubiquitin-conjugated proteins, and as such plays fundamentally indispensable roles in regulating almost all cellular processes. It's got a lot of jobs to do, has to coordinate a lot of substrate selectivity and processing to achieve this regulation. And the inner working of the 26S proteasome is among one of the most sophisticated mechanisms that we have in eukaryotic cells. So questions will be asked at the end of the webinar, but please do send in your questions during the talk. If you have a question, please type it in the question box as shown in the image on the screen, stating who your question is for. Our invited speaker today is Dr. Yudong Mao, who is currently a tenured Associate Professor of Biological Physics and Cryo-Electron Microscopy at the School of Physics and Center for Quantitative Biology at Peking University in Beijing. He's also a senior scientist and principal investigator at the Intel Parallel Computing Center for Structural Biology at the Dana-Farber Cancer Institute, the teaching hospital affiliated to Harvard Medical School in the US. Dr. Mao obtained his PhD in condensed matter physics from Peking University in 2005, then spent 2 years at the National Center for Nanoscience and Technology in Beijing. After that, he then joined the Dana-Farber Cancer Institute as a postdoc fellow in 2007. He then joined the faculty of Harvard Medical School as an instructor in 2012, became the principal investigator and director of the Parallel Computing Center in 2014, and began his independent research lab at Peking University in 2015. His lab is dedicated to the development of computational and experimental methods analyzing highly dynamic protein machines, including the proteasome. And he has pioneered the applications of deep learning in improving cryo-EM data processing pipelines, and has determined numerous high-resolution cryo-EM structures and lots of conformational dynamics of the human 26S proteasome. So I'm really looking forward to hearing about his work. And with that, I would like to hand over to Dr. Mao.
Okay, thank you very much, Helen. And can I start to share my screen? Yes. Okay. Okay, great. Good afternoon to UK people, and good morning for US people. So before I start my short webinar, I'd like to briefly acknowledge and express my appreciation to Professor Robin Harris, who actually invited me to write the chapter for his amazing book named Macromolecular Protein Complex 3, Structure and Function, which is currently shown the slide on the right. And it's actually, he proposed, recommended me to give this webinar. I greatly appreciate the Biochemical Society to give me the opportunity to discuss our most recent studies on proteasome structure and function. So our current slide is just briefly kind of recoup you the idea of where does about of the proteasome is actually it's actually at the end of many, a huge number of ubiquitination pathway. So we know that a proteasome is the major molecular machine that's doing ATP-dependent selective substrate degradation. So this kind of substrate, as shown in the red structure, has to be first modified by ubiquitin, either as monoubiquitin or polyubiquitin, through a cascade of E1, E2, and E3 enzymes. So there's whole lots of studies around E1, E2, and E3, and in fact, the discovery of the ubiquitination pathway centered around E1, E2, and E3 enzymes were recognized by Nobel Prize in Chemistry of 2004 to 3 scientists. So in terms of structure, what are we really curious about and what is really fascinating about this molecular machine is that it's actually combining many different functions. So as I show in the current slide, that as you can see that the proteasome has a core particle that is mainly responsible for breakdown of polypeptides. But we know that protein has to be folded very well to be functional. So prior to polypeptide degradation, and proteasome has to recognize ubiquitinated substrate. So that got to be something called a ubiquitin receptor. So currently we know that there are at least 3 ubiquitin receptors, RPN1, RPN10, and RPN13 in each of the regulatory complex that's attached at either end of the CP or both ends of the CP. And in the meantime, the machine has to unfold the substrate. So it's actually embedded with a AAA-ATPase module that is function as an unfoldase that's hydrolyze ATP and harvest ATP hydrolysis chemical energy to mechanically unfold the substrate. So in the meantime, as you know, that once the ubiquitin has been recognized and its ATPase engaged with substrate, the ubiquitin modification has to be reversed in order to allow processive substrate processing. So as you can see that this molecular machine is an elegant coupling combination of several different machineries. So there are ubiquitin receptor, deubiquitinase, and ATP unfoldase, and protease. So there are many functions, to just name a few, that is actually proteasome is at the center of regulating cell cycle, for example, and some of the tumorigenesis, apoptosis, and immune response. So apparently there's also many other potential functions to be discovered, and it's hardly to be included in one slide. This is just to give you a short kind of recap. So my lab basically we're trying to deal with this highly dynamic kind of complex machinery that's composed of several different or many different enzymes into a hollow enzyme. So we were trying to develop cryo-electron microscopy to visualize complex dynamics. So just to remind you what is the cryo-EM. So basically the idea is we kind of purify the protein and using a cryoplunging flash frozen procedure to freeze them into very thin layer of ice. Normally it's about 20 to 50 nanometer, and then put them into the transmission microscope under cryogenic condition and record images using currently right now called direct electron detector to create these a large number of image stacks. Of course, if you can actually extract all images that's randomly oriented macromolecular particles, you can use computation approach to sort them out into different orientation, and then you can potentially reconstruct this structure. And if you even have more powerful algorithm that can analyze the conformational change, so theoretically you can also solve the complex dynamics. But because of today's webinar is really about proteasomes, so I'm gonna save it for future discussion about the algorithm development in my lab. So we're gonna just jump into what are the end results of all these methodology development. So about 5 years ago, we actually first determined the proteasome structure in the absence of substrate in the presence of ATP. So at that point, the PNAS paper reported with 4 conformations that's spontaneously sampled by the proteasome is called SA, SB, SC, and SD states. So SA state went to about 3.8 angstrom, and it turned out that's a resting state. Then there's only state SD showing the open gate of a CP core particle. So we, in 2018, we kind of published another study by replacing ATP entirely with ATP gamma S. And it turned out these single states, it's not a single state, it's actually composed of at least 3 substates called SD1, SD2, SD3. So these 3 states mainly differ in their ATPase motor module. So how the proteasome regulatory particle actually translocate substrate. And the first is we look at the potential substrate translocation channel in the AAA-ATPase unfoldases, because that's the most interesting part, or say the first part that's going to stably engage with substrate unfolded region. So if you look at this architecture, it's very interesting that each of the ATPase subunit contribute 2 pore loops called pore 1, pore 2 loop. And in the default state, in the resting state, they actually form 2 kinds of superhelical architecture as just waiting there. But however, if you look at the real structures, the central AAA channel is so lateral that such it has to open to accommodate the substrate insertion. So that's going to be showing later. So to briefly summarize the proteasome structure in the absence of substrate, I'm going to use a movie to show it. So this movie basically is assembled from the 3 SD state. So from SD1 change to SD2, then from SD2 change to SD3. So these basically are the 3 states almost equally spontaneously sampled in the presence of ATP gamma S and in the absence of substrate. So it shows that the proteasome is in constant motion, even you don't give it anything, right? So, so that's the work before 2018, but in fact, starting from 2017, try various kind of started all various experiments, try to figure out how to assemble a substrate and capture substrate bound proteasome. So because of limited time, I'd like to jump to the major kind of principle, or say the conclusion is what is shown in the current slide, and we published in 2019. So the really interesting part is the discovery. I will explain a little bit more in detail in next 10 to 15 minutes. Is we found that the ATPase motor is actually almost control nearly everything about the proteasome function, and it's shown as 3 modes. So as you can see, the first dashed line box is the first mode that we potentially is regulating ubiquitin binding and deubiquitination. So as you can see that this mode is featured by kind of ATP hydrolysis in 2 subunits that's just opposite each other. You can see the green molecule is the ADP, and the red molecule here binding to the ATPase nucleotide pocket is the red one is ATP. So you can see the ATP hydrolysis into ADP is kind of taking place in the 2 opposite ATPase, while in the state EB, it's very similar, but it's rotated like counterclockwise by just 1 subunit. And then there's a second mode, and this second mode is different from first one because what we see is 2 laboring subunits kind of hydrolyzing ADP at the same time, or exchanging at the same time, but do not really propagate down to the ATPase ring. So in this case, what we see is actually it's regulating, potentially regulating the translocation of the translocation initiation and ubiquitin release, as well as the safety gate opening. So it's kind of doing multiple things at the same time. So once the core particle gate is open, then we believe, as many other literatures or publications illustrated, that it looks like there's only 1 ATPase is hydrolyzing at a time at a 1. Then is kind of exchanging of the nucleotide. So then these kind of modes rotate around ATPase ring and potentially going around in a circle. But what's interesting for the proteasome is somehow we just didn't see the other 4 possibilities. That's why I put a question mark here. And we would briefly talk about why that's the case. So that's the kind of major discovery, but to really understand why that's the case and associated to the function, we actually kind of have to roll back a little bit. So these kind of mechanism somehow could potentially address nearly all these questions, or at least a majority of these questions. For example, how many conformers can this molecular machine really sample in solution in the presence of substrate? How the ATPase channel is open for substrate engagement, and how does ATP hydrolysis regulate substrate engagement and deubiquitination? And how does ATPase initiate substrate translocation? And how the chemical energy of ATP hydrolysis used to unfold the substrate protein, as well as how is the substrate processing regulated intrinsically, somehow can all be kind of gleaned from what I just showed you, the 3 modes. So if we just change a little bit angle, look at this whole thing, look at if you say let's compare the energy, free energy landscape. Well, the free energy landscape is actually pretty complicated, as we show in the bioarchive paper, which I was not able to talk about here because it's in the process of formal publication. But I draw here as a kind of schematic or conceptual comparison, which is actually easier to read, easier to look at. So without substrate, which I show for ATP or as the red curve, red free energy profile, and for substrate free state with ATP gammas, I show as orange dashed profile. And if you add the substrate, then it's kind of the whole landscape is changed and becomes much more rugged, much more rugged. So you can see for the assay state is kind of split into multiple states. Assay state become EA1 and EA2 and EB state. And also for the other remaining states, they kind of remain, but if you look at our latest publication, it's not just the 4 states here, it's almost 10 states, even more here. So that gives the idea that how this, the landscape of substrate of the proteasome before and after substrate interaction. So then how we can capture them. So we, based on this understanding, we devised the strategies called nucleotide substitution. So that the idea is very simple. We basically feed the substrate with ATP and substrate, then put the timer on, count on 30 seconds. After 30 seconds, we exchange in which that deplete or replace ATP with ATP gammas. So once you replace the ATP gammas, after 30 seconds of degradation reaction, you kind of freeze everything, every potential intermediate states in the middle of reaction. So that's kind of how we did it in terms of trap the substrate engaged human proteasome. Indeed, this strategy works to the effect. It's not just to capture some of the state. To our surprise, is capture almost any states you could imagine, and definitely it's beyond our initial kind of speculation what could be a trap. So then outcome, this is just, I would say a little bit of solid data because it's in 2019, and the manuscript was put out in the bioRxiv 2 months ago, included almost 20 conformers for the RP, single RP and single CP subcomplex. And we're also able to reconstruct the complete pure double cap and differentiate it from the single cap. But all those are somewhat based on the current 7 states. So the 7 states, as you can see, that can be understood into 4 phases. The first step, it's ubiquitin binding, right? It has the protease has to first recognize ubiquitin, then try to do some structure change to prepare for deubiquitination. So that's the second step. And after deubiquitination, it has to smoothly transit into initiation of translocation and try to control the core particles, say, well, open the gate, right? So then after the gate is open, it's supposed to go into a phase that's that is processively degrade a substrate. So what is interesting is that you can still see on this slide, there's a major kind of feature. As you can see, in the absence of substrate, the largest the population is the assay state. But when you add a substrate, the largest the state, which you also see in the particle population, is instead the ED2 state, which is the substrate engaged and in the middle of processive translocation and with the core particle gate open. So that's a very dramatic kind of conformational change, and as well as the dynamic change of the free energy landscape. So these slides just give you a brief kind of overview of 3 of the typical structure in the most important functional step: deubiquitination, the translocation initiation, and processive translocation. And you can see that in the EB state, which is actually doing the deubiquitination work, where we can really see the isopeptide bond between the ubiquitin molecule and the substrate. And in the state EC, you can see that the isopeptide bond somehow has been cleaved. So apparently the ubiquitin has detached from the substrate, while the substrate is kind of going free into deeper in the ATP ring. And in the ED2 state, the ubiquitin on the deubiquitinase is even released. It's gone because we don't need it anymore, right? So the substrate kinds of go into kind of processive translocation modes. And in the meantime, you will see that whole regenerative particle kind of undergo dramatic conformational change we will show in another movie. So just briefly kind of go through some of the local densities where I show in some of the states, for example, the EA actually will reconstruct it to pretty good resolution where you can even see the alanine residue side chain somehow. Also, the ED2 is not too bad. You really can tell many of the moderate size, even some of the small size of side chains. So that most prominently is that we can see the single magnesium ion in the EA state around ATP, and also occasionally can see the magnesium ions even on ADP. So these are the very interesting structures. If you look at it very closely to the ATPase, what the structures show is that in current slide that ADP is that we fix the orientation of ATPase and just look at how ADP or say nucleotide state changes. What is interesting is you can see that in EA1 state, the ADP is at the topmost subunit, and this ADP is kind of the hydrolyzed ADP is kind of moved counterclockwise. Then in the EB state, you'll see the ADP show in the adjacent subunit, and it's moving, and so apparently the ADP state kind of navigating throughout the whole ring. You see the on the rightmost panel, the ED2 state, the ADP is actually rotating to clockwise labor subunit of the ADP in the EA1 state. So what is shown is the 4 cycle of ADP hydrolysis that's navigating through all 6 hexamer subunits. So that's a very interesting observation. And the second important, or say another very prominent observation is that these around the ring nucleotide hydrolysis navigation is strongly coupled with the substrate interaction, as shown in the lowest panel. So you can see that the pore loop is kind of doing the kind of stepping work. Probably it's a little bit kind of, you have to sit down to look at this. You see the green tyrosine residues is the residue that's contacting the substrate main chain from the pore 1 loop. And this green residue, the tyrosine is kind of moving down. You see that in the EC1-2 state, it's moving down by like around 2 residue on the substrate, and it's moving down continuously in ED1 and further down in ED2. So the outcome is during this 4 cycle of around the ring ATP hydrolysis, this green tyrosine residue is moving from the top to the bottom. So and that's solely driven by the ATP hydrolysis. So the current slide, the lower right panel is the diagram showing the pore loop stepping system that I just explained. So let's look at a movie, see how these 7 structure kind of, when they connected together, how it looks like. So this movie was not based on molecular dynamic simulation. It's based on 7 cryo structures and just connect them into a movie, right? So there's no other simulation done for this movie. As you can see, the RPM1 has like 45-degree rotation, then the substrate inserted into ATPase channel, and there's a large rotation, and then release the ubiquitin. Then ATPase move again and drag this whole thing, the red substrate into the CP channel. So that's the 7 structures. So we come back to these 3 modes. Let's go through each mode to look at a little bit more details. So the first mode. The first mode is summarized in the current slide, as I just talked about. In the substrate, AAA-ATPase channel is actually in a closed state, although it's shown in a super helical kind of arrangement that looks like it's ready to accept the substrate, but it's just there's no enough space. So what happens is that we found that the ADP bond to RPT6 kind of has to be released in the state EB in order to open the central AAA channel. So we compare the AAA-ATPase structure between states EB and EA. As you can see, the EB is colored for each of the subunit, and EA was shown as gray. And you can see that the colored structure, particularly the pore loop that's interacting with the substrates, kind of move out, right? Move out a little bit. Then the largest movement is in RPT6. That is almost like it's kind of flipping out. If you go closer to the structure, what happens is in the EA state, you have kind of very tight interface between RPT and its 2 neighboring subunit. But then in the EB state, ADP is kind of released. The RPT6 kind of form a very noticeable marked kind of gap at RPT3, RPT6 interface, and RPT2, RPT6 interface. Then in the later step, you start to see this kind of interfaces kind of close one at a time. Right? So first there's RPT3 to RPT6 interface is closed in the C1 state, while the other interface is still kind of wide open. And in the D1 state, then both were closed. So if you really look at even closer to RPT6 structure, there's a noticeable refolding because there's a, as you can see, that dashed green curve is actually corresponds to an order structure which is well resolved in EB state, but it's but in EA this dashed segment of structure is kind of disordered. So there's apparently after the nucleotide is released from the RPT6, these kind of slightly disorders transit into a well-ordered structure. Also, you can see part of the other structures also refold. So that's a very interesting and prominent feature. Also, as you can see that if you compare the RPT6 ATPase structure between the 5 different states, only this APO state, which is shown as the purple one, where you see a large kind of rotational change between small AAA and large AAA subdomain. While the other four structure are always ADP or ATP bond, they actually is kind of quite comparable, except for the large AAA domain where some of the pore loop kind of looping areas kind of having dramatic refolding. So that's the, and in the meantime, these kind of mode 1 also regulate the ubiquitin recognition. So this is just shows you that two highly occupied ubiquitin recognition sites, one is on RPN1 and near between T1, T2 sites, and the other is between RPN11 and RPT4 and 5 coil. So you can see that in the EA1, there's two ubiquitin kind of hung around the coil. One of the ubiquitins kind of move close to RPN11 in the EA2 state, and it's even more tightly inserted in EB state to prepare the ubiquitin for deubiquitination. So if we rotate the whole structure and almost 90 degrees, then if we magnify the local structure where the isopeptide bond sits, you can really see isopeptide bond is well linked between the substrate and the ubiquitin C terminals. And what's also interesting is there's a kind of unstructured loop in most of the other structures which we call RPT5 N loop. So this N loop is well ordered and it's participating to form a 4-strand beta sheets that's kind of orienting the isopeptide bond for deubiquitination. However, this RPT5 N loop is kind of missing in all other states. And we also know that this RPT5 N loop is highly conserved across yeast to human. So we were scratching our hand when we saw the first structure, why both yeast and human proteasome has to have these kind of N loop that's kind of extra long and kind of doing nothing and not assuming any meaningful structures, but not until we see this EB state and explains why it has to be there. So just briefly mention that in this structure, we also have strong evidence of the catalytic zinc ion in RPN11 active site. So that means that the whole structure is in a state that is fully active, So okay, let's move to the mode 2. So the mode 2, I would just go slightly quicker. So basically what it does is 3 things at the same time. One thing is move the substrate forward by adds together would be, I would say, one step forward, then release the ubiquitin and also trigger the CP gating. So what is about a CP gate opening that is interesting is it's triggered by the C-terminal tail of 5 ATPase. 5 ATPases, but in EA, EB, and EC and ED state, we see different patterns of the C-terminal tail insertion into the surface pockets of the alpha ring, which is actually illustrated in the cartoon in the lower row. That what it is showing is there's one more C-tail inserted into the alpha ring at a time when it moves from one state to the nearest downstream state. For example, the EA1 to EB state, you got RPT2 C-tail inserted. Then you move down to the EC state, you got RPT6 C-terminal tail inserted. Then if you move down to further down to ED state, now RPT1 C-terminal tail is inserted. Then this central gate is open. So that's a very interesting kind of synchronized observation that is kind of shown the proteasome is rigorously regulated by allosteric effects. Then let's look at MOF3. So the MOF3 looks like it's much simpler because what it's showing is 3 kind of neighboring subunit kind of synchronize their nucleotide processing. So as you see in the forest green subunit, this it's undergoing ATP hydrolysis. Then its clockwise adjacent subunit is doing kind of ADP release, and while the further down adjacent subunit is doing ATP binding. So basically what happens, the 3 kinds of nucleotide processing were synchronized around a ring, around half of the ring. Well, one is doing ATP hydrolysis, the second one is doing ADP release, the third one is doing ATP binding. That's a very interesting thing to look at. Of course, this process, which step is faster, which step is slower, it couldn't be seen in this 2 structure. But I can tell you that if you look at our latest about paper, we kind of have the lucky kind of chance to resolve 6 states during single nucleotide exchange, where we start to look at which step is fastest, which step is the slowest, and it tells you more about intrinsic coordination of during the structure transition. So at least from the EB state to ED2, for RPT5, we can really see that a single nucleotide is undergo 3 types of processing. First is hydrolyzed, then ADP released, and then this pocket is rebinds ATP. So it's kind of 4 cycle of ATP hydrolysis and exchange. And then you can measure the conformational change over the whole process. So what we see is that, for example, there's large kind of flipping out, kind of out of the way flipping in the RPT5 ATPase. It's almost flipped like a 30 degree when it transited from ED1 to ED2, which corresponds to ATP binding procedure. So ATP binding is kind of it's kind of doing the things that moving this whole ATP small AAA against the large AAA, or vice versa. So it's very similar to what we see in RPT6 subunit. If you compare the 5 states of RPT5, it's only the apo-like state, which in this case is ED1, that's showing almost 25 degree of rotation between the small and large AAA subdomain, while the other 4 states either bond with ATP or ATP. They show very similar structures. So which means that it's the process of ADP release, or say nucleotide exchange, give rise to the most dramatic intrinsic motion in the ATPase. So that's actually how kind of power stroke were generated, as shown in the current cartoon, that the ones that's releasing the ADP is actually, once it released, it's kind of the whole ATP is going to becomes very flexible intrinsically, so it could rotate 30 degree. Then after this kind of nucleotide release, when it actually binds ATP, is actually going to make the whole ATPase become more rigid, and in which case it only trigger like 7 degree motion overall. But this kind of motion, when they kind of combine together, is enough to generate mechanical force to move the substrate towards the core particles. So let's watch another movie that's showing more intimately how these ATPase kind of doing the nucleotide exchange and drag the substrate forward. Okay, so so this is the EB state, and that's at the very beginning. Then the EC1 state. It's just about to start to move the substrate forward. And EC2, the whole thing kind of move above the core particle. Then the ED1 state. So you can see the pore loop actually grab these substrates very tightly, but in the EC1, EC2, there's two pore, one loop dissociated from the substrate, but in both ED1 and ED2, only one pore one loop dissociated from the substrate. So that's connect the whole thing together and look at more how the substrate were translocated. Okay, so it's the same as the previous movie that we did not do any molecular dynamic simulation. We just connect these structures together and just let them to play. So the current slide basically kind of expands the idea of the three modes, and because the previous we only showed ATPase, but in fact the change of the ATPase conformation is kind of doing things in both ways. It strongly affects the core particles conformation as well as the surrounding lead subunit. So the lead subunit actually, you can see that the lead subcomplex sitting above the ATPase module is contacting the ATPase ring in different sides. So that kind of interaction probably is kind of provide a strong constraint on how these ATPase ring kind of rotate doing ATP hydrolysis around a ring, and why we kind of see ED1, ED2 states more frequently, while not see other four potential states clearly. It's probably because it's making these two states much more stabler through the lead-based interaction, while the other four states kind of conjectively disfavored, but probably still sampled, but it's very quickly, extremely quickly, that we probably won't be able to capture them at all at this point, unless we have some other strategies. Last slide is trying to kind of stimulate some of the thoughts is what's the future? What's the immediate future and the longer term future? So we kind of look at three different directions. One is that how to characterize conformational landscape of ubiquitin signals required for proteasome targeting, and try to understand how these ubiquitin signals interact with the ubiquitin receptors in the context of proteasome. And that's very important and very difficult job to do. Also, in fact, if you look at these two things, there's many things could happen in between protein ubiquitination and proteasome-mediated degradation. And also, since now we can actually image this very sophisticated molecular machinery at atomic level to extract its dynamics information, then we want to ask a question about how to harvest, how to convert these dynamic informations into therapeutic or drug discoveries. So previously, we only used kind of static structures to design drugs, but we know that many drugs that interact with some of the immediate state, transient state, or just change the energy landscape. So if you have many structures, if you say you have 60 structures of a proteasome, how that's going to impact your drug discovery. I think that's a lot of things to digest and a lot of information to really make sense. Okay, so briefly acknowledge my group member and three heroes. First, Yuanchen Dong is now already, he was a postdoc in my lab a few years ago, and he's now a professor at Institute of Chemistry of Chinese Academy of Sciences. And Xu Wenzhang is now a postdoc in my lab, and he was my graduate student who did amazing work with Yuanchen Dong on the 7 structures. And Zhaolong Wu is my current grad student who developed the alpha query 4D that allow us to expand the 7 structure into more than 60 structures. And also I'd like to briefly acknowledge the Professor Mark Kirshner's group. And really collaboration with Mark's labs really got all this kind of proteasome started. So that's I'm really appreciative of that, very thankful for this collaboration. Thank you very much. That's all about today's talk. Any questions?
Okay, wow. Thank you very much, Yudong. That was an extraordinarily impressive tour of the proteasome. We have a couple of questions that have already come in. And please, if you do have questions, please type it into the questions box. So we have one from Sergio Ribeiro, who is asking, are there any cell stress molecules or external toxins that could bind to the RPN proteins and therefore affect substrate recognition, folding, or degradation?
Well, I think there's well, I couldn't precisely answer this question. That's because there's a large number of network. I would say a large number of protein networks is actually regulating the proteasome function upstream. So there are proteins apparently involved in prepared substrates. For example, the CDC48 in yeast and P97 in human. Kind of some of the substrate won't be able to have this initiation region ready for proteasome. Then the CDC48 P97 is kind of prepared for it, right? So there's also other, I would believe that some of these, right now there's also, besides the extrinsic ubiquitin receptor like RAD23AB in human, ubiquitin, there's 4 types of ubiquitin that's apparently kind of very responsive to some of the cellular stress. But also though that some of the E3, like for example, UB3C, 4-5, they also kind of been involved in this misfolded protein response that kind of got involved in the function, regulating function of proteasome. So yeah, that's a very good question. Has actually matched my last slide that there's a whole lot of things going on between protein ubiquitination and proteasome degradation. So many proteins actually indirectly or directly involved in the regulation of proteasome. And also we know that the proteome looks like kind of constantly being chemically modified post-translationally in response to certain cellular stimuli. And when not fully understand it, some of the phosphorylation really stimulates selectivity, and some of the kind of modification instead kind of break it down. So a very good question, I would say, about those things. And the question actually represents probably a whole new area in the future to be explored. And right now, I think it's just many possibilities. Yeah.
So we have another question from Betul Karademir, who is asking, do you think there's any situation where these 18 subunits are separated from the catalytic part?
can you restate you mean which subunit is separated from catalytic part?
So they're asking the 18 subunits in the regulatory proteasome, are they ever separated from the catalytic part? I think that's the question.
Yes.
Yeah.
Oh, yes. Yeah, they are separated from the proteolytic active sites, which is actually sequestered, concealed inside the beta ring. It's the very deep down. Let me just show you the slide. Sorry, you cannot see slide, right? Okay, I'm just gonna describe it. It's actually done in the very deep part of the core particle inside the beta ring. So it's well separated from the regulatory particle, that's for sure. But of course, the regulatory particle itself has kind of deubiquitination active sites and ATP hydrolysis active sites also. But apparently these are the enzymatic sites in a regulatory particle is clearly allosterically coupled to the proteolytic sites inside the core particle. And that's actually a major conclusion of the bioarchive paper. We really see some of the coupling going on there. And there's also, I would tell you, there's a series of paper from Louis Kay's lab in Canada. They were using AMR show these kind of long range global couplings all around the proteasome, while they were showing it using archaeo proteasome. But I think it's quite true for the 26S.
Okay, great. And I think maybe along similar lines, Pedro Fernandez has a question about whether a double capped proteasome can take two substrates at once.
Yes, of course. Yeah.
Yes,
that's been seen. Yeah, that's been seen.
Excellent, thank you. And I think Sergio has another question, that since the proteasome is really dynamic, do you have any data about the movement of different proteasome subunits while the proteasome is actually being formed or assembled or dismantled? So you're looking at it as a whole holistic thing, right? But have you got any data when it's in process, if you see what he's asking?
So does it mean that in the process of assembly or in the process of functioning? In the process of assembly, we do capture regular particle before it actually assembled and the core particles. So that was a molecular cell paper. And what is interesting is we found that if the whole regular particle was not assembled and the core particle, its ATPase is sample multiple conformations. It's actually sample continuous distribution of conformations. It doesn't stay still at all. Yeah.
And so I think,
yeah.
Okay, I think I actually had one question, and then I was going to ask Jack's technical question. Just out of curiosity, how many ATPs then do you need to destroy a substrate? Is it one per residue? With that stroke that you're talking about, every ATP hydrolysis is moving it down a substrate. Do we have a sense of how energy intensive destruction of 100 amino acid substrate is?
Well, very good question. I can break it down to this. Why ATP hydrolysis based on the structure looks like it's kind of moved the substrate forward by two residues. So we can kind of calculate probably if you say 100 amino acid, I would say at least 50 around ATP. Yeah.
Wow.
So that's kind of, yeah. But we know there's also, you probably need a few more ATP to get it started, because the starting point is consuming more energy. As I show in what I'm talking about is the mode 3. The mode 1, actually, you move it once, you take 2 ATP. In mode 3, you move it 2 steps, you only take 1 ATP hydrolysis. So, yeah. So a little bit more than that.
And so Jack Stubbs would like to know how you made the movies that you showed us of your time-resolved cryo-EM.
Well, how we made the movies? First, you have to do the data collection, solve the 7 structures, right? Then it becomes very simple because the chimera has a simple function. If you have a 7 structure that is so close, it will simply interpolate, connect these 7 structures into a movie. You don't need, because the structure is so close, so they can just interpolate the trajectory of the atoms. So that's how it was made. It's not made by molecular dynamic simulation. Just give UCSF chimera the coordinates, then you would just create a movie for you.
Excellent.
In minutes, very quick.
Excellent. All right, so that takes us to just gone 3. So I would like to thank everybody for attending, and I would really like to thank you, Yudong, for a superb presentation. I really enjoyed that. If you're not done with your questions yet, you can always continue the conversation online. You can follow the Twitter accounts that are shown here. Okay, and so just as a few final wrap-ups, we do welcome any suggestions that you might have for future topics, speakers to feature in this webinar series. So if you've got any ideas, we invite you to submit a proposal for a webinar, and you can find more information at the website shown here. Please do join us for the next one in the series, which will be called Novel Advances in Signaling, Next Generation Approaches, and that takes place on the 10th of March at 3 o'clock GMT. And that one is part of our dedicated early career researcher series, and it will feature presentations from 4 early career researchers working in the field of signaling, and will be chaired by Dr. Amy Lewis, who is a postdoctoral research associate at Queen Mary University of London and a member of the Biochemical Society Early Career Advisory Panel. You can find out more about this and register on the website. And so obviously this is an extraordinary time for all of us, and it's more important than ever that we stay connected to each other, if only virtually. So it's also an exciting time to join the Biochemical Society as we're looking to expand the online activities. So I've been a member of the Society for many years now, and I take full advantage of the support that is offered, particularly to attend meetings. And as I have become more established in my career, the freedom and the opportunity to organise meetings with society support, not just financial, but also administrative support. You can also apply to host seminar series, and essentially the society facilitates and enables communication of science and formation of networks across multiple platforms. And you can join this community of researchers and specialists and take advantage of the key benefits, which include discounted registration fees, access to a wide range of grants and bursaries, personal online access to their journals, and more. So please do visit the website to find out more. Thank you very much again for joining. It was a pleasure to chair this session. I hope you have a good remainder of the day, whatever time it is with you.
Thank you very much.